The Disinfectants Lab. In this lab, we use TSA trypticase soy agar plates, which contain soybean salt, agar, and casein. We need 5 TSA plates because we will be testing 5 different bacteria. We will also need discs, sterile discs, for the disc diffusion method that will be demonstrated. These discs will be dipped into different kinds of disinfectants, and we will test to see the efficacy of these disinfectants on different bacteria. We will use iodine, 10% chlorine bleach, 70% isopropyl alcohol, 3% hydrogen peroxide. These are the disinfectants used. The bacteria used include Staph aureus, E. coli, Bacillus cereus grown for 2 days, Mycobacterium smegmatis grown for 2 days as well, Bacillus cereus grown for 7 days in TSB broths. Notice all of them are in trypticase soybean broth, liquid broths. The first thing we want to do is proper labeling. We use a Sharpie and we label our plates in this manner. We put some dots, 4 dots, about 1.5 centimeters away from the edge of the plate, and you can see the ruler here, and we label 4 dots, 1 for alcohol, 1 for bleach, 1 for iodine, and 1 for hydrogen peroxide. We do this with all the plates, all 5 of the plates. This is our E. coli plate that we've labeled, our Bacillus cereus plate, our Bacillus cereus 7-day plate, our Mycobacterium smegmatis plate, and our Staph aureus plate, all labeled properly. Now for the procedure. We will be using these sterile plastic pipettes or dropper pipettes. We carefully open the bacterial culture and take some of the bacterial culture. Carefully place the tube back into the rack. Place a couple of drops onto the plate. That's, of course, labeled for that bacterium. We pick up a sterile swab and we swab the bacterial culture all over the plate, making sure we don't leave any gaps. We turn the plate and keep swabbing, covering the whole surface. We call this a lawn plate. We cover the whole surface of the plate. It is very important you cover the whole surface of the plate. Make sure we dispose of anything that has bacterial culture on it in the hazard trash. I will repeat the procedure for the next bacterium. We use a sterile dropper pipette. Carefully, trying to prevent contamination, pick up our bacterial culture sample. Place a couple of drops in our plate. And notice our plate here is open and the lid is aside for demonstration purposes. Here is the sterile swab. We cover the plate fully, and again, having the plate open like this may introduce some contamination from vents, things in the air. but it's the best way I can demonstrate to you a lawn agar plate. We repeat this procedure for all the other bacteria. So now for the next part of the procedure. We have our tweezers, forceps, in ethyl alcohol, and then we make sure that we sterilize them in the Bunsen burner flame. We pick up a sterile disc and we select one of our disinfectants. We will actually place a drop or two of this disinfectant onto the sterile disc. Sometimes we may want to dab any excess on the paper seen on the aluminum foil, and then we place our disc right on to the labeled disinfectant area. So if this was bleach, we put it on the dot that is labeled B for bleach. We move on to our next disinfectant, making sure we use Aseptic Technique. Our tweezers must be in alcohol and also sterilized in a flame. We pick up our second antibiotic, drop a drop onto our diffusion disc, place it on the appropriately labeled dot. Make sure we tap it a little bit so it kind of sticks to the surface of the agar. We continue with our third disinfectant. Sterilize our forceps, tweezers. Pick up a sterile disc. Drop a drop of disinfectant. May want to dab off some excess. And then place it on the appropriately labeled dot. Make sure you tap it in order for it to stick on the surface of the agar. Always sterilize the forceps. And now we are ready for our fourth disinfectant. Here's our disc. One drop of disinfectant. And we place it on the appropriately labeled dot. And since this is the iodine, it should go on the dot that says I. We have finished our first plate. We have plated the bacteria and we have placed the discs with the disinfectants onto the first plate. I will demonstrate one more time, placing the discs with the disinfectants on the plate. Pick up a disc and our disinfectant. Put a drop onto the disc. Dab off any excess and place it on the appropriately labeled dot. Tap it, make sure it sticks to the agar. Pick up a disc, drop the disinfectant. Notice we lost that disc. Sometimes that happens. We want to make sure we pick up a new one, a sterile one. Place our disinfectant, dab off any excess, place it on the appropriately labeled dot. Moving on to disinfectant number 3. Same procedure. Pick up a disc. Oh, we lost that disc. Now it's contaminated. We Pick up a disc. A drop of disinfectant. Dab off any excess. And we lost that one again. So we get a sterile disc. Place our disinfectant. Dab off any excess and place it on the appropriately labeled dot. We've got one more to go. Always using the aseptic technique, sterilizing our tools. So our last disc, this will be the iodine. Place a drop of iodine disinfectant or antiseptic onto the sterile disc and place it onto the appropriately labeled letter I. We repeat for all the other plates, for all the other bacteria. After we are done, we incubate all the plates at 37 degrees Celsius for about 24 to 48 hours, and we come back and record results. The photos will be provided for recording results to the students. Thank you for watching this video.